Good morning.
Good morning. What's all this? Your results. Congratulations to all. I'm confident you'll all achieve your goals. So, young man, what are your aspirations?
I want to do robotics, ideally from the Bust Institute in the US.
And you, young lady?
Sir, I love kids and I would love to learn pediatrics. I feel Boston is the best place for that.
Sumit, what about you?
Back home to India for medicine.
Good. I'm sure you'll all achieve your aspirations, be it in India or anywhere. Keep in mind, your first challenge is the entrance, whether it's IIT, JEE, SAT or NEET. You have to learn from the masters. Brilliant Qatar is the passport to your success.
We undertake coaching for regular and repeat batches and tuition for board examinations. Brilliant Education Centre, Doha, Qatar.
Hi all, welcome to Brilliance classes. Today I am going to start with some important questions from biology of grade 10. So many important areas are there which is expected in the coming exam and all that I will be discussing in today's session. Keep on commenting your answers. I will be discussing the topic along with some MCQ question. Now let me start with the first question. Which one is the correct summary of equation of photosynthesis? You know what is photosynthesis? The process by which the green plants prepare their own food using carbon dioxide, water in presence of sunlight and chlorophyll. And remember, it's an anabolic process. Anabolic means what? It's a constructive process where you can see small particles will be combining in order to form a complex particle. So what are the smaller particles that are combining and what is the complex particle that is formed and all? Keep on commenting your answer. You may be knowing this. So here you can see here 6 CO2 plus 12 H2O. This is the smaller particle 6 CO2 plus 12 H2O that will be giving C6H12O6, that is glucose. It's the complex particle that has been produced releasing oxygen and water. So this is the right equation for photosynthesis. Remember some more points related to photosynthesis, that is it's an anabolic process. What is anabolic process? It's a constructive process. Second, you should remember it's a redox reaction. Redox reaction means there is reduction and oxidation. So what is actually reduced and what is oxidized? Remember, carbon dioxide is actually reduced into glucose. And you can see the water will be undergoing oxidation. Water is undergoing oxidation in order to release oxygen. One more question, from which molecule the oxygen is liberated in photosynthesis? So if you take carbon dioxide and water, you should understand the oxygen in photosynthesis is coming from the water molecules. It's not from CO2. So here, remember it's an anabolic process, it's a redox reaction, carbon is reduced and water is oxidized. And where is this oxidation of water taking place in order to release the oxygen? Can you please comment where the oxidation of water is taking place? Abhinav, Niranjan and all already have answered, it is D. So here, oxidation of water will be taking place in the chloroplast, especially in the grana. In the grana only water will undergo splitting in order to release the oxygen. The process is called as photolysis. So photolysis of water will be taking place inside the grana. Hope you remember the site of photosynthesis and all. So which is not the site of photosynthesis? Here, main site of photosynthesis is chloroplast, that is located in the mesophyll. And even the stomata will be having chloroplast. So that is also a location where photosynthesis can occur. But in the epidermal cell, you should understand there is no chloroplast. So there is no chance of photosynthesis in the epidermis. Hope you know the structure of a dicot leaf. So let's recap the structure of a dicot leaf. If I take the cross section of a dicot leaf, it will be having an upper epidermis as well as a lower epidermis. Like this will be the structure of a dicot leaf. You can see there is an upper epidermis and a lower epidermis. Lower epidermis will be having the stomata that contains the guard cell. And in the midrib of the leaf, it will be having xylem and phloem. It's covered by the bundle sheath. Now, in between the upper and lower epidermis, there is palisade as well as spongy mesophyll. Like this compactly packed mesophyll tissues will be coming. That compactly packed mesophyll tissue only, we call it as palisade mesophyll. And here, less compactly packed mesophills are coming. That is actually called spongy mesophyll. So you should understand, inside this palisade as well as spongy mesophyll, they will be having chloroplast. And the photosynthesis will be occurring inside the chloroplast, that is coming in the palisade as well as the spongy mesophyll. So here each chloroplast should be getting water from the xylem, and they also should be getting carbon dioxide from the atmospheric air, which will be diffusing through the stomata and reaching all the chloroplast. So site of photosynthesis, if it is asked for the exam, the main site of photosynthesis is green parts of the plant. In the green part itself, it will be mainly confined to the leaf. In the leaf, it will be mainly taking place in the mesophyll. In the mesophyll, chloroplast. That is the location of photosynthesis. Now, we can divide the photosynthesis into two, that is light reaction and dark reaction. So, in the chloroplast, the structure you may be knowing, there is an outer membrane, there is an inner membrane, and they will be having coin-like structures that are called thylakoids, and they are joined to each other by the stroma lamella. So in this thylakoid or grana, the light reaction will be taking place, which will be releasing out energy currency like, so using sunlight, the light reaction will be taking place in the grana, because you should understand the photosynthetic pigments are located here. So trapping the light energy, they will convert it into chemical energy like ATP and NADPH. That will be released into the stroma part of the chloroplast. In the stroma, you can see the carbon dioxide will be fixed by the most abundant enzyme in the plant world, Rubisco. Here, you can see 6CO2 will be fixed in order to form C6H12O6. So this reaction only we call it as dark reaction. So photosynthesis we can convert, or we can write it in two steps. One is light reaction, and where is the light reaction occurring? It's mainly occurring in the grana, where you can see there is energy production like ATP as well as NADPH. These energy currencies are produced in the light reaction. Second, you can see there is dark reaction. Even you can see there is photolysis of water that will be taking place in the light reaction, which will be releasing out electrons, oxygen and the H+. Then there is dark reaction. Dark reaction will be taking place in the stroma part. In the dark reaction, you can see using this energy from the light reaction, the carbon dioxide will be fixed as glucose. The carbon dioxide will be fixed as glucose in the stroma part. So this is how photosynthesis is taking place. 1 mark and all, if it is coming, you can directly write. There it is an anabolic process where the green plants prepare their own food using CO2 and water, and they will be converting it into glucose, releasing oxygen. So where is the oxygen liberated? If the question is coming, it is in the grana. From which molecule the oxygen is liberated? The answer is water. There is photolysis of water that will be taking place in the light reaction in the grana. Hope you understood this much. Now let's move to the next question. Which one is correct? Light reaction occurs in the stroma, that is incorrect. Just now I said light reaction in photosynthesis, it's not occurring in the stroma, it's occurring in the grana. Whereas dark reaction is what is occurring in the stroma, because stroma part of chloroplast is having enzymes associated with photosynthesis. Now second statement, light reaction occurs in grana and ATP and NADPH are formed. So that is actually correct. Next one, stroma, the dark reaction occurs. Dark reaction is not directly light driven, but it is dependent on products of light reaction. That is also correct. You should understand it will be depending on the products of light reaction. The product of light reaction is ATP and NADPH. Using that only the dark reaction will be occurring in the stroma side. So 2, 3 and 4 are actually correct. Next one, which one of the following is the correct definition of photosynthesis? You should answer. You may be knowing this. Yes. There is conversion of light energy from the sun into chemical energy. So in photosynthesis, light energy is converted into chemical energy. It's not electrical energy or anything. So here light energy is converted into chemical energy like ATP and NADPH. Using that, you can see carbon is getting fixed. So that first statement is actually correct. Many are answering. Very good. Abhinav. Now next one. why the leaf to be tested for starch have to be boiled in alcohol? So before starting this, let me discuss about the iodine test for detecting the presence of starch in the leaf. You know that the product of photosynthesis is glucose, but in the mesophyll, they are actually getting stored as starch. That is also a point. So in the mesophyll cell, there is chloroplast. The exiting sugar after photosynthesis is glucose, but you should understand this glucose will be joined in the mesophyll cell in order to form a complex sugar, that is starch. So starch is the storage form of sugar in the plants, whereas if it is to be transported, the glucose will be combining with its isomer, that is fructose, in order to form sucrose. This is the transport form of sugar in the plant, which will be loaded into the phloem from the mesophyll cell. You should understand phloem is the transporting tissue, conducting tissue that is coming in the plants. If they want to transport something from the leaf to other part of the plant which require food, that will be done by the phloem only, and the transport form of sugar in the plant is not glucose. The transport form of sugar in the plant is sucrose, which will be loaded into the phloem. Hope you got this much. Now, here we will be performing starch test using iodine. So what are the procedures that you should do is, first pluck out the leaf, put it in boiling water. After that, you have to put it in a test tube which contain alcohol and again boil it. After that, remove it and you can put it in the water, wash it nicely, keep it in a watch glass, put some drops of iodine. If it is turning blue-black, understand the presence of starch. It is turning blue-black. Now, here, why the leaf to be tested for starch have to be boiled in alcohol? The main purpose is to dissolve the chlorophyll. The main purpose is to dissolve the chlorophyll. That is why we actually, to remove the chlorophyll only, we are actually boiling it in alcohol. Now, next question. Given below are the steps to be followed for performing starch test on a green leaf. Boil the leaf. So you have to arrange it in right order. So please comment what will be the right order of the different steps associated with this. So what is the first step? Boil the leaf in water. That will be the first step. Then boil the leaf in alcohol. Just now I have said that is the second step. Then wash it. Then you have to dip the leaf in iodine solution. This will be the right order of the steps associated with the experiment to show starch is present in photosynthesis using iodine test. Now next one, a portion of each of the 4 discharged leaf of a plant was covered with paper strips of various kinds. The plant was exposed to sunlight for 5 hours. Thereafter, the strips were removed and the leaves were tested for starch in the covered portion. Which one out of the 4 leaves gives starch test in the covered portion? You may be knowing about the experiment. Light is essential for photosynthesis. First of all, you have to keep the plant in a dark room for 48 hours or 72 hours, 2 to 3 days, so that all the starch present in the plant will be utilized. After that, take out the plant, put it in sunlight. And on one of the leaf, keep a black strip or a colored paper or strips you have to keep, so that that portion will be deprived of sunlight and remaining portion of the leaf will get sunlight. So after 5 or 8 hours, pluck out the leaf and perform the iodine test as we discussed earlier. So here, those leaves which were covered by black strip, green paper, white paper, etc., it won't turn blue-black in that covered region. So those area which was covered by the strip, it won't be turning blue-black. But if you are putting a transparent paper, surely that region will turn blue-black because they will be getting enough sunlight. Next one, what are stomata responsible for? You know about stomata, especially found in the upper and lower epidermis of the plant. In monocot, it is found in upper epidermis and lower epidermis, but in dicot leaf, it is found only in the lower epidermis, and it will be very less in number in the upper epidermis. So here, main function of stomata, just now we have discussed what is the main function of stomata. Hope you got this structure of leaf. Through the stomata, only carbon dioxide enter into the leaf, or from the xylem, the water will be exiting out of the leaf as water vapor, that is transpiration. So transpiration to bring a cooling effect to the leaf and all, it will be occurring through the stomata when the water is lost as water vapor. And here you also can see carbon is entering through this side only, and even oxygen exiting also. So exchange of gases and transpiration is the most important function of stomata. Hope you are getting it. So functions of stomata, if you are taking, the functions will be including transpiration and exchange of gases. Then structure of stomata can be asked for the exam. You should be knowing how to draw it. So stomata, when you are drawing, the inner wall should be thick and outer wall should be thin. Like that only you should draw if it is coming. So inner wall of the stomata is thick and outer wall is very thin. And at the center you can see they will be having a large vacuole. A large vacuole will be present at the center, and in the periphery they will be having a nucleus. In the periphery they will be having a nucleus, and so many chloroplasts also will be distributed in the peripheral cytoplasm. So you have to mark it, and at the center the gap that is opened, that is actually called stoma, and the surrounding epidermal cell we call it as subsidiary cell. So in vicinity of the guard cell, the surrounding epidermal cell only we call it as the subsidiary cell. So subsidiary cell and this guard cells together along with the stomatal aperture, that is actually called as stomatal apparatus. That is called as stomatal apparatus. Hope you got the answer. Now next one, select the incorrect statement. In dicot leaf, stomata is present more on lower epidermis. That is actually correct. In monocot leaf, stomata is equally distributed on upper and lower. That is correct. In dicot leaf, more stomata on upper. That is wrong. Now next question. When should the leaf be plucked in order to prepare a temporary slide of leaf peel? So if you are taking a dicot leaf, you have to take the peel from the lower epidermis, and you have to take a fresh leaf only for doing this experiment. Now next one. Which part of the leaf is used for making a leaf peel mount in dicot? Of course, the most of the stomata is located on lower epidermis, so you have to prefer a peel that is taken from the lower surface. Next one, which hormone promotes cell elongation by promoting water intake and increase cell elasticity? So cell elongation, you may be knowing about the bending of plant towards cell elongation, especially for promoting the water intake and increase cells elasticity. Cell enlargement, cell elongation, the answer is A only, auxin. Hope you remember about the phototropism. In phototropism, the plant was bending towards the direction of light because of auxin only. Auxin, those who are migrating to the shaded side, there the cells were elongating. That was making the plants to bend towards the direction of light. Hope you got it. Yes, Abhinav. So the answer is A. So understand, in a plant, auxins are produced at the apex. It will be diffusing downward. When it is diffusing downward, the cells in the shaded region, suppose light is from here, you should understand auxin will come to this shaded region. In the shaded region, the cells will start elongating. So cells in the shaded region, if it is elongating, slowly you can see the plant will start bending towards the direction of light. So cell elongation is done by auxin only. Hope you got it, Abhinav. Now next one, carbohydrate in the plants are stored in the form of, remember, it is stored as starch. And also remember what is the transport form of sugar in the plant. Transport form of sugar in the plant is sucrose, and it is transported by which conducting tissue? Phloem. Hope you remember the function of xylem and phloem. Xylem will be carrying water and minerals from the soil to all the upward parts of the plant, whereas the phloem will be carrying the food from the leaf to all the growing regions in the plant that is not able to perform photosynthesis. Now next one, which of these juices is secreted by pancreas? You know that into the duodenum there is hepatopancreatic duct joining. This is the pancreas, and there will be a pancreatic duct here. So what are the things secreted by the pancreas? So this is the glands associated with the pancreas. So this exocrine part of pancreas will be secreting pancreatic juice here, and it will be including the protein digesting enzyme like trypsin, chymotrypsin, etc. Like that, many enzymes will be secreted. Even they will be secreting the starch digesting pancreatic amylase also, even pancreatic lipase for digesting the oil. So this much things will be secreted. Pepsin is by the stomach, that you know. In the stomach, the gastric glands will be producing the pepsin, whereas bile is secreted by, here the bile will be coming from, so this is the bile duct, and what I am going to draw with pink, that is the cystic duct from the gallbladder. So here only the bile is actually stored and released into the duodenum portion. Whereas the bile will be coming from where? The bile will be mainly coming from the liver only. So liver will be having, so backside of this understand there is liver. The hepatic duct will be coming from the liver like this. That from the hepatic duct of the liver, they will be releasing the bile that is stored in the gallbladder, later released by the gallbladder. So bile is by the liver. So the answer is trypsin. Trypsin is actually produced in the pancreatic juice. Yes, the answer is A. Now next one. Yes, Abhinav, got it. Now match the column 1 with column 2. So you should be knowing the difference between, the capillaries, veins, aorta and wall of the vessel. Can you please match it? So capillaries, you should understand, it will be having only one cell thickness. Narrowest blood vessel will be the capillary. It will be having only one cell thickness. Whereas veins in that will be having valves. And aorta, they will be having elastic artery so that they can pump the blood under high pressure and all. And wall of the vessels will be having smooth muscle. So smooth muscle 3142, 3142. 3 is the answer. Hope you got it. Yes, Rahul Vijay, that is the answer. Now next one. So this difference can be asked for the exam. The difference between artery, vein and capillary. Arteries are having thick walls, vein thin wall, one cell thickness in the capillary. Then artery always take blood away from the heart, veins always bring blood towards the heart. Like that also you try to remember. So arteries and vein, please try to remember this one. Arteries are always bringing the blood to, the veins are always bringing the blood towards the heart. Whereas arteries will be taking the blood away from the heart. Like that you try to remember. Now next one, blood carry carbon dioxide in 3 forms. The correct percentage of carbon dioxide in these forms are, try to understand, after aerobic respiration in the cell, a lot of CO2 is accumulating in the cell that can be toxic to the cell and it has to be removed out of the cell. So especially this CO2 will be diffusing from the cell to the capillary. And in the capillary, they can be transported in 3 forms. From capillary, they will move to the veins and they will be joining the vena cava and coming to the right side of the heart. So in which all form the CO2 is transported from the cells to the heart and heart to the lungs. So you may be knowing the double circulation pathway and all. So here 70% will be, 20 to 25% will be transported by the RBC. Around 23% as carbaminohemoglobin, 70% will be as bicarbonate, 7% as the carbonic acid in the plasma. So these are the transport form of carbon dioxide in the plant. Got it? Yes, Rahul Vijay, that is answer. Now next one. So here you should understand, from the cells CO2 will be entering into the venules, vein and all, and it will be reaching the heart. So right atria it will be reaching, then to the right ventricle. From there it will be going to the lungs. So it is taken away by pulmonary artery. Hope you got it. So from the cell, CO2 is taken away by the veins, which will be joining the largest vein. What is that? Vena cava. And after entering right side of the heart, it will be going to the lungs. And in the lungs, what is happening is there is alveoli. So in the alveoli, so you should understand, alveoli, they are also having one cell thickness. It is made up of squamous epithelia only. Then it will be having a surrounding. So here the pulmonary artery will be coming with the carbon dioxide containing blood, and it will be joining the narrow capillary. It will be joining the narrow capillary on the surface of the alveoli. They will be joining the narrow capillary. In the capillary only you should understand there is diffusion of carbon dioxide. So carbon dioxide will be entering to this side, and you also can see oxygen will be entering into the blood. And this oxygen will be taken away by the only vein that carry the oxygenated blood, that is pulmonary vein. So this is what is happening in the exchange of gases. Yes, Abhinav, hope you got it regarding this one. Hope you are taking the points and all. Now next one, figure shows human urinary system structure. Labeled A to D, select the option which correctly identifies them and gives their characteristic and function. So D portion, so in the kidney the outer portion is called as cortex, and they do not contain any part of nephron is wrong. They actually contain cortex region in the kidney will be having. So if I am taking the kidney like this, you will be knowing a concave side and the convex side. So here only the renal pelvis will be coming in the kidney. So here renal pelvis will be coming. In the renal pelvis attached will be a lot of renal pyramids. Like this you can see a lot of renal pyramids will be attaching. And here only you can see there is a lot of nephrons that are coming. So a lot of nephrons will be coming like this, entering here. So the nephrons, this much portion around the Bowman's capsule, PCT and the DCT, all these are coming in the cortex region. So this statement is wrong. Nothing of nephron is present in, no part of nephron is actually coming in the cortex and all. If it is stated, that is wrong only. So here, then second one, adrenal gland located at anterior part. So on the top portion of the kidney, there is adrenal gland, which will release the emergency hormones like adrenaline. You know, when is the adrenaline rushing out? In three situations, when you are feared of something, when you go for a fight or a flight, in these conditions, your body will be emitting adrenaline. Adrenaline, when it is rushing out, so many things will happen. That also questions can be there. You should understand the heartbeat will keep on increasing in a feared situation. And your pupil will start dilating. So just imagine a dog is chasing you. What is the situation and what all things will happen? The heartbeat will keep on increasing. Breathing rhythm also will keep on increasing. And you can see there is a lot of blood circulation to the muscles and all. And the dilation of pupil. All these are the things associated with adrenaline. So where is adrenal gland located? It's located above the kidney. In that adrenal medulla part will be producing the hormones like adrenaline and noradrenaline. So this is actually correct. Then pelvis, medulla, that all I am not doing. Now next one. So what are the three steps in urine formation also? Let me just recap. During urine formation, you should understand the blood that is to be purified, especially the renal artery entering into the kidney, they will form arteriole and they will be joining the capillary. In the capillary only, the first step will be taking place, that is called glomerular filtration or filtration under pressure. So when our blood is actually circulating, so when our blood is pumped through these capillaries and all, there will be a lot of pressure in that. Because of the pressure, so many waste particles and some good particles will be diffusing into the Bowman's capsule. So like this, so many good particles and waste particles will be diffusing into the Bowman's capsule. The process is called glomerular filtration. First step in urine formation is glomerular filtration. From this you can see so many useful particles will be again sent back into the blood. That is called tubular reabsorption. That is called tubular reabsorption. So urine formation process, if coming for the exam, you should write three things. One is glomerular filtration or filtration under pressure. It happens when the blood is flowing through the glomerulus under high pressure. In that you can see so many useful particles and waste particle will be coming into the Bowman's capsule. From that useful particle will be returned back into the blood. That is tubular reabsorption. And again whatever waste is left will be again put back into the nephrons. That is called tubular secretion. So this is what is happening in urine formation process. Now next one, identify A, B, C and D. I am expecting the comments for this question. You should be able to identify what is A, what is B. I got the answer. Can you please comment fast? What is A? Rahul, Gayatri, expecting for some comments from you people. So here A represent pons varoli, that is a part of hindbrain. Then B is the second largest part of the brain, that is cerebellum. Second largest part of the brain. Partially it is overlapped by, yes, very good, you are answering. So A represent pons. First answer was given by Abhinav. Abhinav has answered first. So A represent pons, that will be controlling or regulating the breathing. Then cerebellum, that is the second largest part of brain, that is B. Okay, so many started answering also. Okay, good. Then D represent thalamus and C represent cerebrum. So these are the different parts: pons, cerebellum, thalamus, pons, cerebellum, Pons, cerebrum, cerebellum. So here which answer I can give? A is pons, B is cerebellum, then C is cerebrum, pons, cerebellum, cerebrum, thalamus. Okay, 4 is the answer. Got it? Okay, many has commented. I think so many started commenting first itself. You got the answer. So which is the largest part of the brain? You already know that will be controlling the memory as well as they will be controlling the movement, the voluntary action, involuntary action, memory, as well as vision, hearing. All these are coming in the cerebrum. Then cerebellum, second largest part of the brain, they will be controlling the posture and movement of a person. So although for the movement the signals are coming from the cerebrum, it's coordinated by, the coordination of movement is actually done by cerebellum, and it will be attaching to the pons, and the brain stem will be formed by medulla oblongata that will be connected to the spinal cord. It's not thymus, it is thalamus. It's a part of forebrain. Abhinav, forebrain has 3 parts: cerebrum, thalamus, hypothalamus. Like that 3 part is coming in the forebrain, and in that hypothalamus will be controlling the urge for eating, thirst, etc. So thirst, hunger, all these are associated with hypothalamus. Thalamus is the gatekeeper of cerebrum. They will control which all nerve impulse should go to cerebrum and exit the cerebrum. That is under the control of thalamus. It's not thymus, Abhinav, please note it, it is thalamus. Now next one, a person was unable to walk normally after taking alcohol. Which of the following part of the brain probably get affected by alcohol? Can you tell? What can be the reason why he is not able to walk properly? He is actually moving a lot. He is going to fall and all. Which part of the brain is getting affected? Or the person is trying to ride a cycle but not able to ride the cycle properly. He is falling. Why? Which part of the brain is affected? Rahul, Vijay. What can be the answer? Yes, first answer by Hiba. Hiba, you are correct. Hiba's answer was cerebellum. Yes, cerebellum is the right answer, because posture and movement or the coordination of movement is mainly done by cerebellum. Even though the movement signals are starting from the cerebrum, in which way it has to be coordinated so that we can get the correct posture and all, that is under the control of cerebellum. So cerebellum is the answer. Very good. Okay, it's okay, Rahul. Happens. Now next one, select the correct option. You know this is associated with respiration. Now let's have a discussion about respiration. What is respiration? The process by which organic molecules are broken in a cell in order to produce some energy is called respiration. Cellular respiration, what I mean. So the most common respiratory substrate that is available is glucose. It is 6 carbon. You should understand whether in plant cell or animal cell, this glucose will be broken down in the cytoplasm to form 2 units of a 3 carbon component. That 2 units of 3 carbon component is pyruvic acid. And here the process is called, what is the process called? The process is called as glycolysis. Then you can see the pyruvic acid will be taken into the cell organelle mitochondria. In our case and all, it is taken into mitochondria. In the mitochondria, this 3 carbon containing pyruvic acid, it will undergo oxidation. So 3 carbon containing pyruvic acid, 2 units are there. It will be broken in the mitochondria to release CO2 as well as the water, and it will also produce energy. In aerobic respiration, you should understand they produce around 38 ATP molecules. So these all things happens in the mitochondria. Steps involved are Krebs cycle, link reaction, like that many things are there. So this is the basics only. So aerobic respiration is completed in, starts in cytoplasm but completes in mitochondria. And how much energy is produced from 1 glucose? 38 ATP is produced from 1 glucose. But in anaerobic condition, in the muscles and all, in anaerobic condition, try to understand this will be converted into lactic acid. That may result in muscular cramps. It will be producing lactic acid. It can cause muscular cramps. Whereas anaerobic condition in yeast and all, it will result in the formation of ethanol and carbon dioxide, and it yield only 2 ATP. So energy production in anaerobic respiration is very less. Now tell which is the right option which I can go for. So here C6H12O6, that is glucose, and this process require oxygen also. You should understand it will be requiring oxygen also. So 6O2 giving 6CO2 plus 6H2O plus energy. Pyruvate or pyruvic acid, both are same only, Rahul. Don't get confused if I have written pyruvic acid. It is pyruvate. Pyruvate or pyruvic acid, both are same only. Now, here for this oxygen will be supplied by that only we have discussed about the double circulation and all. You should understand from the lungs the oxygenated blood is coming to the left side of the heart. From left atria it moves to left ventricle. From there it is picked by the aorta, from aorta artery, then it will be joining with the capillary, supplying the oxygen to the cell. Using that oxygen only aerobic respiration take place, which will again result in CO2, which will be taken away by the blood, the veins, and again vena cava. Like that it will keep on rotating. So hope you got a continuity. Okay, Rahul. Now next one, temperature in the scrotum necessary for spermatogenesis or sperm formation. You know that in males, the testis is located outside the abdominal cavity in a pouch-like structure that is called scrotum. And the main function of the scrotum is what? What can be the main function? Can you please comment? What is the answer for this question? Yes, first of all, the answer was given by Rahul itself. They will maintain a temperature 2 to 3 degrees Celsius less than body temperature, because in that temperature only there can be sperm formation. In some people, testis will not descend down from the abdomen, from the birth itself. In that case, the sperm formation is not possible. The condition is called cryptocridism. It's not required for our syllabus. Just I am telling, just to make you understand what is the function of scrotum. So in our body temperature, the spermatogenesis cannot occur. It can occur if there is a temperature less than 2 to 3 degrees Celsius less than our body temperature. Then only sperm formation is possible. Now next one, fight or flight reaction cause activation of. You already know the answer. So which hormone is associated with fight, flight, fear, etc.? Can you please comment which hormone is associated with fight, flight and fear? Let me check who is going to answer first. The hormone associated with fight, flight and fear, the triple F hormone. Okay, Rahul said the first answer. That is 3. Abhinav also. So here adrenal medulla, adrenal gland, if you are taking, it is having 2 parts. The center portion is called adrenal medulla and the peripheral portion is called adrenal cortex. In that adrenal medulla only will be releasing out the emergency hormone, that is adrenaline. So adrenaline will be secreted by adrenal medulla. Now next one, pancreas, you already know it is producing the blood sugar, it will produce hormones regulating the blood sugar level and all. And parathyroid gland, that will be actually controlling, it will be regulating calcium level in the body. So antagonistic hormone is parath hormone, the antagonistic hormone will be the calcitonin. So calcitonin and parath hormone, they will be working opposite in action in order to control the calcium level. Then that is not required at all. Now next one, hormone associated with ripening of fruits. You might have learned about the plant growth hormone, phytohormone. You might have learned about the growth hormone as well as inhibitor hormone. 3 growth hormone you might have learned, auxin, gibberellin, cytokinin, and 2 are inhibitors, ethylene and abscisic acid. In that the main hormone associated with the ripening of fruit, they are actually released as a gas only. So if you keep some ripened orange as well as unripened banana together, you can see within the next day the banana will also start ripening, because from this orange they will start secreting this gas, that is ethylene. The answer is ethylene. Very good, many are answering the question. Ethylene is the answer, that is the gaseous plant growth, the plant hormone which is associated with the ripening of fruits. And you may be knowing ABA. ABA, what I have written is abscisic acid, the stress hormone. Learn it as the stress hormone produced in the plant during emergency situation. So in the plants and all, during emergency condition, that means if the temperature is very high and all, you should understand the plant cannot perform a lot of transpiration. If they lose all the water, they will start wilting. So if the water status in the plant is very less, and if the outside there is a lot of stress like high temperature, further transpiration is not possible. So what they do is they produce the emergency hormone abscisic acid. So functions of abscisic acid, closure of stomata, in order to reduce transpiration. Then they will also do all the things associated with reducing the loss of energy. So they will make the seed as well as the bud to enter dormant stage. And they will also leads to, or they will promote abscission of ripened fruits as well as the old leaf, etc. So that much energy won't be lost. These are the functions of abscisic acid. Auxin already you know, cell elongation, cytokinin cell division. So these are the hormones that are coming in our topics. Now next one, the number of chromosome in a diploid parent cell. Diploid parent cell, let's take it as ourselves, 46. We have 46 chromosome. How we can maintain? So our male parent as well as female parent, that will be diploid with 46. So the question, the offspring of a particular species remain constant due to. That means how we can tell our progeny, whether it is male or female, they are still having 46 chromosome. How? The answer is the chromosome number will become half in the gamete. So the male gamete, it will be having 23 chromosome, and the female gamete also will be having 23 chromosome. So here remember a kind of cell division associated with the halving of chromosome number. The cell division is meiosis, or you can learn it as reductional division. Reductional division is associated with halving the number of chromosome. Fertilization will again restore the diploid condition. Fertilization will be bringing the diploid condition back. This is the things associated with the gamete formation and fertilization in diploid organism. Always remember, diploid organism, to restore the ploidy, the gametes are always haploid, and this haploid condition of gamete is due to which division? Meiosis. So here it is halving of the chromosome number in the gamete. Okay, Anna has answered and deleted. Then the answer is actually halving the number of chromosome, not after zygote formation. First you answered is correct, halving the chromosome number in the gamete. After zygote formation, it will be again becoming diploid only. Now next one, which of the following is the correct sequence of reflex arc? When you touch a hot object and all, what is happening you already know. So let's discuss about reflex action. So here there is a receptor. So when you are touching a hot object and all, there is a stimulus. That stimulus will make the receptors to produce what? Chemicals. These chemicals will be attaching to the sensory neuron. They will be attaching to the sensory neuron, and sensory neuron will be carrying a nerve impulse to your spinal cord. So inside the spinal cord, you should understand there is another neuron. Usually when you touch a hot object, from the receptor, sensory receptor, the nerve impulse should go to your spinal cord, then the brain. But here what happens is, to prevent unloading of all the nerve impulse and all, here the spinal cord itself will be managing it with the help of another neuron. That neuron is actually called as relay neurons. So from the relay neurons, you can see the message will be passed. So from the sensory neuron, the message is going to the spinal cord, and in the spinal cord there is a relay neuron that will be giving the messages to which neuron? It will be giving the messages to the motor neuron. So motor neuron will be carrying this message. So this is the motor neuron, and this is the, so this will be coming like this, and this represents the sensory neuron which is carrying the sensory impulse. So relay neuron connect them, and this message will be coming to the effector. Effector means it can be muscles and all. For example, if I touch a hot object, message is going from my hand to the spinal cord, then the motor neuron bring the message back to my muscle, that is the effector. So I push it back on a sudden. So this is going to bring the response. It is going to bring the response. Hope you got this much. Now next question. So let me check the reflex action. Which of the following is the correct sequence of reflex arc? Receptor, motor neuron, that is wrong, sensory we should get. Receptor, sensory neuron, spinal cord, relay neuron, motor neuron, effector. So we got the answer, answer is B. Many started commenting now itself. Niranjan, okay, right answer. So it is B. Now next question. The growth of tendril in a pea plant is due to. You should understand, when they are getting something to stick on. For example, pea plants and all, when they are growing, when they are getting something to stick on, they will start growing around it. So how they will start growing around it? So you should understand the touching area. So those area the cells are touching, that cells will not be growing, but the opposite cells will start elongating. So this area the cells will be normal touching area. Auxin will migrate to those area where there is, the hormones will migrate to those area which is not touching on the wood and all. So that cells, when they start elongating, slowly they will start encircling around. So rapid cell division in tendrillar cell that are away from the support, that is making them to grow or making them to curl around this touched objects. So this is an example of thigmotropism. Tropic and nastic movement, learn what is the difference. Growth dependent movement is tropism, growth independent movement is nastic movement. So growth dependent movement, they will be bringing growth in the plant and they will also bring movement. Nastic movements you know about thigmonasty, photonasty, etc. So in that there will be just movement, but it will not be bringing any growth. Touch me not, that is thigmonasty's example. Blooming of flower and closing, that is photonasty's example. Response is due to sunlight. Then here you should understand it's actually bringing growth. Phototropism, geotropism, chemotropism, all these you can see growth dependent movement. That difference you should be able to write well. Now moving to the next one. Okay, so many answers were coming in that. Anne Mary, Abhinav, right answer. Now choose the incorrect statement about insulin. You already know what is the function of insulin. It will mainly be produced by the pancreas in order to reduce the blood sugar level. So in the blood, the glucose level when it is increasing, suddenly you should understand the pancreas will be producing. The pancreas, there is endocrine part. Endocrine part of the pancreas will be secreting. This is the exocrine part. Whereas endocrine part of the pancreas will be secreting insulin. And what happens is, these sugars will be absorbed by the surrounding adipose tissue or it will be taken up by the liver. So two things can happen. This sugar will start moving to the adipose tissue or it will be taken into the liver where they are joining to form glycogen. So glucose will be joining to form what? Glycogen. This is how insulin is working. They can decrease the blood sugar level in two forms, either making the cells to uptake it or it will make the liver to uptake it and convert it into a polysaccharide, that is glycogen. Hope you got it. Now let's come back to the question. Choose the incorrect statement about insulin. It's produced from pancreas endocrine part. It regulates growth and development of the body. No, this is associated with actually growth hormone. Then it regulates blood sugar level. Insufficient secretion can cause diabetes. Diabetes mellitus is actually caused by insufficient secretion of insulin. So these are the points associated with insulin. Now next one, dwarfism in human beings is caused by due to the deficiency of growth hormone only. Excessive secretion can actually lead to gigantism and very less secretion can lead to dwarfism. Then next one, growth of pollen tube towards the ovule, that is an example of chemotropism. You should understand ovule will be secreting some chemical. Because of that only the pollen will start forming the pollen tube, that is passing through the style and reaching the ovary, entering into the ovule. Hope you got it. Yes, right answer. Now next one, in which order do these events occur in human nutrition? So in human nutrition, first is intake of food, that is called ingestion. Then second is digestion. After digesting the food, you can see they are absorbed in the villi, that is called absorption. Then in the cells they will be used for different purpose, main metabolic activity, that is assimilation, and what is left is given out also, that is called egestion. So these are the various steps associated with human nutrition. So if you are getting a question regarding nutrition in amoeba, same things you should remember. Amoeba will be engulfing a food particle by producing, it will be engulfing a food particle by producing a finger-like projection. That finger-like projection is called pseudopodia, and they will cover it in order to form a food vacuole. They will cover it in order to form a food vacuole. Inside the food vacuole only, this food particle is present. Then second thing that happens, so this process is ingestion. Second thing that happens, so this process is phagocytosis. Amoeba producing finger-like projection, engulfing the food particle, and they produce a food vacuole. into the food vacuole, they will start releasing the digestive enzyme, and slowly you can see this will be broken into small particles, and the small particles are absorbed into the body. So there is ingestion, digestion, absorption, and the absorbed particles are used for energy production or other activities and all, that is assimilation. Then whatever waste is left here is removed out of the body, that is called egestion. So when you are getting it, write that 5 steps itself. Now moving to the next one. What is the correct route of blood flow in humans? So double circulation, a simple diagram, hope you can remember it nicely. So respiration we already discussed. A glucose is broken down into carbon dioxide, water and energy. This carbon dioxide, if accumulating in the cell, it can be toxic to the cell. So what happens is they are getting excreted out. So carbon dioxide will be picked by the smallest vein, that is called venules. So first it will be entering into the smallest blood vessel, that is capillary. From there it will move to venules, and this venules will be joining where? The venules will be joining the veins. Then the veins will be joining where? The largest vein that is entering into the right side of the heart, that is called vena cava. So I am drawing it little bit faster for understanding the topic. So please try to recollect the points. So here there is a tricuspid valve, and here there is a bicuspid valve that is coming in the heart. This is the 4 chambers of the heart. This is the right atria, this is the right ventricle. So the deoxygenated blood from the vena cava enters into the right atria, then it moves to the right ventricle. From the right ventricle, you should understand, this blood will be taken away to the lungs, and to the lungs it will be taken away by the pulmonary artery. That is the only artery that is carrying deoxygenated blood. From the lungs, CO2 is given out when you are exhaling, and oxygen is taken in when you are inhaling, and then you can see this oxygenated blood is brought to the left atria by the pulmonary artery, the pulmonary vein, the only vein that is carrying the oxygenated blood. From left atria, it will be going to left ventricle. Also remember, the thickest wall is of the left ventricle. From the left ventricle, oxygenated blood is taken away by the largest artery, that is aorta. Then it will be joining the small artery, then arteriole, which join the capillary. Later the oxygen is supplied. So this is double circulation. You should understand the same blood is rotating through the heart twice. So same blood is rotating through the heart twice. So here the blood that is coming on right side, after going to the lungs, it's returning to the left side. Like that, this is called as double circulation, which is seen in birds and mammals. Double circulation is seen in birds and mammals. So left auricle, left ventricle, lungs, that is wrong. Second one, left auricle, left ventricle, that is also wrong. Then right auricle, right ventricle, lungs, then from there left auricle, left ventricle. So D option is actually correct. Hope you got it. Can we draw this diagram instead of the one given in the textbook? You have to explain it. If coming for the exam and all, you have to explain it nicely. Then when you are drawing this heart, don't draw for the exam. You have to draw it exactly. For your understanding easily, only I have explained this structure. Circulation part and all okay. Then heart, you have to replace the other one. Drawing the arteries upside and the veins like that. Shall I proceed? Yes, that's it. Now next question. Organism of a higher trophic level which feed on several types of organism belonging to a lower trophic level. If an organism is actually feeding on a lot of organism, it will be not a part of food chain, I should tell. It's an interconnected food chain. So here the answer is it's a food web. It will be a food web, not the food chain, it is a food web. Hope you got it. So interconnected food chains are actually called as food web. And where you can see there is sequence of arrangement of organism in an ecosystem through which the energy flow in an ecosystem, that is called food chain. Interconnected food chain is called as food web. Each position of an organism in a food chain is called as what? That is actually called as position of an organism in a food chain is called trophic level. Now here, excessive exposure of humans to UV ray, this one you should tell. What will happen? It can be damage to immune system. There can be skin cancer. So this is the answer. Next one. Okay, in a food chain, okay, first of all, answer is given. I have, okay, this is also an expected question. Biomagnification. The accumulation of some toxic chemical in the highest trophic level in a food chain is actually called as biomagnification. So biomagnification, remember, it is the accumulation of toxic chemical in the top level. So if you take the trophic level 1, 2, 3, 4, the top level it will be accumulating. For example, DDT and all, if it is sprayed on the grasses, slowly you can see the concentration of this chemical will be increasing in the top person coming in the food chain. That is called biomagnification. Very good. So many are answering. Hiba, Abhinav, Anna, very good. You all have done the right answer. Now next one, what can be the genotype of a progeny when a cross is conducted with capital T, capital small t, when a cross is conducted with capital T small t and small t small t. The question is regarding genotype, not the phenotype. Okay, how you are getting D? You should be able to understand how to take the gamete. Here they can form two gamete, capital T small t, here only small t. So here they can form capital T small t individual, that is tall, small t individual also. None other genotype is possible. So simply don't answer, check the question carefully. What I have written is a test cross. Crossing a dominant heterozygous with a recessive parent is test cross only. Hope you got it. So the answer is B. Don't simply jump and answer due to anxiety, check it carefully. This can be a mistake otherwise. Okay, now got it? Okay, sure. Now let's move to the next one. Pathway of sperm in a female reproductive system. So female reproductive system, when you are taking, you know there is a pair of ovaries that are coming in a female reproductive system, and there is a fallopian tubule that will be coming. Then from the fallopian tubule there is uterus, then there is cervix, then the vagina. So a sperm, so like this they are connected by ligaments also, don't forget that. So many ligaments will be attaching to this ovary, to the uterine wall. So here the pathway of sperm, if you are taking, it will enter through the vagina, cervix, uterus, fallopian tube. So this is the pathway of the sperm. So it is vagina, cervix, uterus and fallopian tube. That is the pathway of a sperm inside the female reproductive system, and the fertilization will be taking place. Please comment fast. Where is the site of fertilization in the female reproductive system? Which is the site of fertilization? All have answered A. Okay. Vagina, cervix, uterus. Okay, both are same only. Okay, A and D. There was a typing mistake. I agree with that. A and D was correct. Okay, one person has Rahul, Vijay, Abhinav. Okay, good one. Now, my question is like this. Where is the fertilization occurring? Which is the site of oviduct? Niranjan answered first. Oviduct is the site of fertilization or fallopian tube. Now, next one. Factors responsible for rapid spread of bread mold on slices of bread are. So, you know about the spore formation in rhizopus. Learn that diagram nicely. So rhizopus is the bread mold. They will be having some filament-like structures that is coming here. That filament-like structures that is coming here is called as rhizoid. And it is hyphae. Tip of the hyphae, they will be producing sporangia. Tip of the hyphae, they are producing sporangia. And inside the sporangia, you should be knowing they will be having so many spores. That spores are actually called as sporangiospores. The spores are called as sporangiospores, they are thick-walled. So tip cell of the hyphae, hyphae means the branch of a fungi, that is called hyphae. The tip cell undergo repeated division in order to form so many spores, that is called sporangia. So they will slowly break and they will release these sporangiospores outside. Whenever they are falling again on good media, they will start germinating. So here it's in large number, availability of moisture and nutrients in the bread that will promote the. So it is A and C. No. Okay, A and C both are same only. So the answer is 1 and 2. Hope you got it. Now next one. The many projections on the wall of small intestine, the function is to. What is the main function? The main function of villi is to. Yeah, we actually came across that. Now the next one. Next one, come in fast. This already you be knowing what is the function. The main function is it increase the surface area. So function of villi, understand, small intestine will be having some finger-like projection. This finger-like projection is called villi, and the cells on this villi will be again producing, the cell membrane of the villi will be again producing some structures that is called as microvilli. That is called as microvilli. So here there is villi and here there is microvilli that will be coming. So if asked about how the small intestine, the surface area is increased, you have to write about villi and microvilli. These two structures actually increase the surface area for absorbing the food particle. So in that there is lacteal. Lacteal means lymphatic vessel will be coming inside. Into that only the digested lipids are uptaken. So digested lipids are uptaken into which vessel? Lymphatic vessel. So remember like this, digested lipids. So if this question is coming, you have to write this much. Digested lipids will be going into the lymphatic vessel. Lymphatic vessel. Whereas surrounding that you can see there is blood capillaries that are coming. So like this, so many blood capillaries will be coming around the lymphatic vessel. So into this blood capillaries also, so many digested proteins and carbohydrates will be taking. Can you see? So like this, the blood capillaries also will be spreading like this. Into the blood capillary you can see there is absorption of digested proteins, that is amino acid. Amino acids as well as sugars will be, digested sugars will be absorbed into the blood capillary. So hope you got this much regarding villi. Bile is synthesized and stored respectively by. You should understand bile is stored, it's produced by the liver. From the liver, the hepatic duct will be forming like this, a common duct. Then later this bile will be stored inside the gallbladder, which will be released through the bile duct that is joining the hepatopancreatic duct. Hope you can understand. So bile, you should understand, it is produced in the liver, stored in the gallbladder. Whenever required from the gallbladder, it will be released into the C-shaped portion of the small intestine. What is the C-shaped portion? That is duodenum. Like this it will be secreted. Hope you got it. So bile is synthesized by liver and it is stored in gallbladder. Both are not in gallbladder, both are not in liver. Salivary amylase digest. You should understand, starch is a polymer of glucose. Like this, many glucose will be joining in order to form starch. So in the buccal cavity or in the small intestine, you should understand amylase. In the buccal cavity it is salivary amylase that is produced by the salivary gland. Salivary amylase will be breaking this bond here. So like this 2,2 glucose will be formed. That 2,2 units of glucose that is formed is called as, that 2,2 units of glucose that is formed is called as maltose. This is the digestion of starch that is taking place. So they convert starch into maltose. So that's all about some important questions that are coming in bio. Now coming to one more question, with that I am stopping this session. Dash is associated with emulsification of fat. Emulsification, you should understand, there is large fat droplets broken into small fat droplets. It's done by bile, that is released by, bile is secreted by gallbladder. So I already said liver actually release it, but it is stored and released by the gallbladder, in which part it is actually released in the small intestine. So in the small intestine, large fat globules are broken down into small fat droplets by the bile. This process only we call it as emulsification. So bile is associated with emulsification of fats. Then after emulsification only, it is acted upon by lipase enzyme, which will digest them into fatty acid as well as glycerol. That's all about today's question discussion. bolus, the food that you are actually taking in, the digested things from the stomach or else from the buccal cavity, that is bolus. Shall I proceed? Now, I'm ending up today's question discussion. Hope you got all the important concepts in the 5 biology chapters. We were just going through some important topics, that is some short questions that we are expecting for the coming exams and all the best to all the students who attended this session and the classes at Brilliant, and all the best to all of you. Okay then, bye.
Good morning.
Good morning. What's all this? Your results. Congratulations to all. I am confident you'll all achieve your goals. So young man, what are your aspirations?
I want to do robotics, ideally from the best institute in the US.
And you, young lady?
Sir, I love kids and I would love to learn pediatrics. I feel Boston is the best place for that.
Sumit, what about you?
Back home to India for medicine.
Good. I'm sure you'll all achieve your aspirations, be it in India or anywhere. Keep in mind, your first challenge is the entrance, whether it's IIT, JEE, SAT or NEET. You have to learn from the masters. Brilliant Qatar is the passport to your success.
We undertake coaching for regular and repeat batches and tuition for board examinations. Brilliant Education Centre, Doha, Qatar.